Bismillahirrahmanirrahim. Good morning, respected chair, organizers, faculty members, and participants. My presentation is titled as Synthesis of Nigella sativa L-based nanoparticles of zinc oxide, copper oxide, and their nanocomposites for evaluating their role in chickpea growth under salinity stress. This study focuses on the green synthesis of metal oxide nanoparticles using Nigella sativa seed extract and evaluates their potential as nanobiofertilizers to improve chickpea growth under salt stress. The presentation is organized into the following section. First of all is the introduction, and we will see the introduction about the nanotechnology, that it deals with the materials at the nanoscale, generally between 1 and 100 nanometer, uh, to generate materials with size-dependent physical, chemical, and biological properties because they can improve the nutrient availability, they can increase the uptake efficiency in agriculture, and they support the plant growth under the stress conditions. Metal oxide nanoparticles such as zinc oxide and copper oxide nanoparticles are especially important because zinc and copper are micronutrients which are involved in enzyme activity, photosynthesis, antioxidant defense, and plant metabolisms. And we have used the Nigella sativa seed extract to synthesize these nanoparticles. Why we have selected the Nigella sativa? Because it is rich in natural antioxidants and bioactive compounds. It contains thymoquinone, thymol, flavonoids, and other secondary metabolites that may act as reducing, stabilizing, and capping agents during the nanoparticle synthesis. Using plant extract makes the synthesis process more eco-friendly, cost-effective, and biologically compatible compared with the conventional chemical synthesis method. Therefore, Nigella sativa was selected as a green source for synthesizing copper oxide, zinc oxide, and their nanocomposites. Here is the problem statement that chickpea is an important legume crop in Pakistan and plays a key role in food security and soil fertility. However, salinity stress is a major agricultural problem that reduces germination, plant growth, biomass production, and final yield. Salt stress mainly causes the ionic imbalance, oxidative stress, and reduce the water uptake in the plants. Therefore, this study was designed to explore whether green synthesized zinc oxide, copper oxide, and their composites could reduce the harmful effect of salinity and improve the chickpea growth. Following are the main objectives, which include a synthesis of the nanoparticles, their characterization, and then to evaluate their effects on the chickpea growth, physiological attributes, and salinity tolerance, and then to compare the effect of these nanoparticles as nanobiofertilizers under the salt stress. This slide summarizes the overall methodology of the study. The work started with the preparation of Nigella sativa seed extract, then zinc oxide, copper oxide, and zinc oxide, copper oxide nanoparticles were synthesized using the green synthesis protocol. After synthesis, nanoparticles were characterized to confirm their formation, structure, morphology, and particle size. The synthesized nanoparticles were then applied as foliar treatments to chickpea plants grown under normal and salinity stress conditions. Finally, morphological and biochemical parameters were recorded and analyzed. Nanoparticle synthesis strategy includes for the zinc oxide nanoparticle, zinc acetate was used as the precursor salt along with Nigella sativa extract. For copper oxide nanoparticles, copper sulfate was used as the copper source. For zinc oxide, copper oxide nanocomposites, both zinc acetate and copper sulfates were combined with the plant extract under controlled heating and stirring condition. The plant extract helped in reduction and stabilization of the nanoparticles. The final products were washed, dried, and stored for characterization and plant application after the process of calcination. Multiple characterization techniques were used to confirm the nanoparticle formation, including the UV visible spectroscopy to observe the optical properties and absorption peaks. FTIR helped to identify the functional groups from plant extract involved in capping and stabilization. SEM was used to observe the surface morphology and particle arrangement. DLS provided information about the hydrodynamic size distribution in suspension. And finally, XRD was used to study the crystalline structure, phase nature, and lattice characteristics. Healthy chickpea seeds were used for the plant growth. They were surface sterilized and sown in the plastic pots containing the fertile loamy soil. Plants were grown under controlled conditions and they were monitored regularly. Then salinity stress was applied using the two concentrations of sodium chloride, which include the 100 millimolar and 150 millimolar. After salinity treatment, nanoparticles were applied as foliar spray at different concentrations. This design helped to compare the normal growth, salt stress plants, and nanoparticle treated plants under the salinity stress. As you can see, the different chickpea seeds they were sown in the plastic pots containing the fertile soil, and growth of the chickpea plants is shown. Then morphological and biochemical parameters were measured. Morphological parameters included the shoot length, the root length, shoot fresh weight, shoot dry weight, root fresh weight, root dry weight, number of leaves, and number of lateral roots. The biochemical parameters included the catalase activity, hydrogen peroxide activity, and peroxidase activity. Together, these parameters provided a clear picture of plant growth performance and antioxidant response under the salinity stress. Statistical analysis, we have used the randomized complete block design for this experiment. The whole experiment was carried out in triplicate, and after obtaining the data, it was analyzed with the help of Microsoft Office Excel. Here are the results. First of all, we have divided actually results into two major parts. The first part focusing on the nanoparticle characterization and other part focused on the chickpea performance. So first is the characterization. We have used the UV visible spectrophotometry, and this slide represents the UV visible spectra of the zinc oxide nanoparticle. The absorption peak observed in the spectra indicate the formation of the nanoparticles. On this slide, the same spectra is shown for the copper oxide nanoparticles and zinc oxide, copper oxide nanoparticles, which also shows the spectral features indicating the combined presence of both metal oxide nanoparticles. Then FTIR analysis was performed to identify the functional groups. The spectra shows the peaks corresponding to the different chemical groups which were derived from the Nigella sativa extract. These functional groups may be involved in reduction, stabilization, and capping of the nanoparticles. Now, this is the spectra for the copper oxide nanoparticle, and this FTIR spectra is for zinc oxide, copper oxide nanoparticles. Then scanning electron microscopy was performed to examine the morphology of synthesized nanoparticle. The images show that the nanoparticles formed visible surface structure and aggregates at the different magnification. So here is the confirmation of the formation of the zinc oxide nanoparticles at 5 micrometer and 10 micrometer resolution. In this diagram, you can see the formation of the copper oxide nanoparticles, and here is the nanocomposites. Then DLS was used to estimate the hydrodynamic size distribution of nanoparticles in suspension. This technique measures the Brownian motion and helps to determine the particle size behavior in liquid medium. This spectra confirmed that the synthesized particles were distributed within nanoscale range, although some aggregations may occur due to particle interaction in the suspension. And this information is important for understanding the foliar splay behavior and the nanoparticle availability to the plant. So here are the results of the DLS spectra for the copper oxide nanoparticle, and this shows the DLS spectra for the zinc oxide, copper oxide nanoparticles. The X-ray diffraction analysis was performed to study the crystalline structure of the nanoparticle. Diffraction pattern shows here it supports the formation of the zinc oxide, copper oxide, and zinc oxide, copper oxide nanocomposites. XRD gives the information about the crystal structure, phase identity, and crystallinity. The presence of characteristic peaks confirms that the synthesized materials had crystalline metal oxide nanoparticle structure. Then nanoparticle were foliarly spread for, and then morphological parameters of the chickpea plants were recorded. This slide shows the effect of the nanoparticles. This is the visual response of the chickpea plant under different nanoparticles and salinity treatment. Salt stress generally reduces the plant vigor, but nanoparticle treated plants showed the comparatively improved growth. The image here provide the visual support that foliar application of nanoparticle help to reduce the salinity damage. Among the treatments, higher nanoparticle concentrations, especially the 500 millimolar, which is shown in the part C, appear to produce the stronger growth responses. And this figure is for the zinc oxide nanoparticle. Same was observed for the copper oxide and for the zinc oxide copper oxide nanoparticle. Then shoot length and root length were observed. Shoot length is an important indicator for the plant growth under the stress condition. Nanoparticle application improved the shoot length compared with the salt stressed plant. And it suggests that these nanoparticles could help to support the cell expansion, nutrient uptake, and overall vegetative growth under salinity stress. Same results were obtained for the root length. That increase in the length of root was observed when 500 millimolar copper oxide at 500 millimolar stress condition when nanocomposites or the zinc oxide or copper oxide nanoparticles were sprayed. Then we have also observed the shoot biomass, which is the fresh weight of the shoot. And it also reflects the above ground plant biomass. It reduced the shoot biomass, but nanoparticle treatment improved the biomass accumulation. As you can compare the results of the control with the nanoparticle. This improvement may be due to the better photosynthetic activity, improved nutrient availability, and enhanced stress tolerance. Among the treatments, the higher nanoparticle dose showed a stronger positive response, indicating dose-dependent improvement in the shoot growth. Similarly, shoot dry weight was calculated and under the salt stress condition, and it is found that its dry weight or its growth is restricted due to the osmotic stress and ionic toxicity. Nanoparticle treated plants showed the improved weight with the salt stress plants. Same is the case with the root fresh weight and root dry weight. Then impact of the nanoparticles on number of leaves under the salt stress was calculated. The leaves are important for photosynthesis, while we have also observed their effect for the number of lateral roots, which are important for absorbing the surface area of the plant. And under salinity stress, plants usually produce the few leaves and weaker root system. Nanoparticle treatment improve both leaf number and lateral root formation, suggesting better plant recovery and improved growth architecture under the salt stress. After this, we have also checked the effect of the nanoparticles on biochemical parameters of the chickpea plants. Biochemical analysis focused on the antioxidant related parameters, including the catalase activity, hydrogen peroxide, and peroxidase activity. Salinity stress increases the reactive oxygen species, which can damage the membranes, proteins, and cellular structure. Nanoparticle application enhanced the antioxidant response in chickpea plant. As you can see, increased catalase activity, increase in the hydrogen peroxide activity, and increase in the peroxidase activity, which indicates that treated plants had a stronger defense system against the oxidative damage caused by salinity. So here are the key findings of our study, which are that characterization confirmed the successful synthesis of zinc oxide, copper oxide, and the nanocomposites. Foliar application of nanoparticles reduced the harmful effects of the soil stress and improved the plant growth. The highest tested dose, 500 millimolar, showed more prominent improvement in growth and antioxidant activity. And overall, copper oxide nanoparticles showed the most efficient responses compared to the zinc oxide and copper oxide zinc oxide nanocomposites. So the future perspective, in the future, this research can be extended to the field level trials to confirm the performance of these nanoparticles under natural environmental conditions. Further studies should optimize the nanoparticle dose, its timing, and spray frequency to ensure the safe and cost effective application. It will also be important to evaluate the nanoparticle effects on soil microorganisms, plant metabolism, and long term environmental safety. This work can support the development of targeted nano biofertilizer strategies for saline affected agricultural region. These are few of the references, and thank you. With this, I conclude my presentation. Thank you for your attention. I would be happy to answer any question regarding the synthesis characterization, plant treatment design, or result of this study. Thank you.